chromosomal inheritance and different chromosomal syndromes, and genetic testing and genetic diseases. There's a little section also on cloning, so reproductive cloning and what that looks like, and some bioethical implications. All right, so outline of what we're going to cover. I have flying in chromosomes and chromosomal inheritance, meiosis, reproductive cloning, chromosomal syndromes, a few of those, genetic testing, how do we do that, and genetic diseases. So we'll go through sort of systematically through those different topics. Animals, plants, fungi, bacteria all have offspring that look similar to their parents. We're animals, right? We're vertebrates, we're mammals. We look like our parents. We don't look exactly like our parents, but we look similar, right? We inherit a suite of genes and traits, right, that make us look similar. In some cases, some of these, like certain types of fungi and bacteria, and even certain plants, they actually are their parents, okay? How they reproduce is by cloning. All they do is pinch off or bud off a section of new cells that are literally just mitosis of themselves, okay? So they're exactly the same, they're cloning. There's like little mini, almost jellyfish-like animals, they're called hydra, and they just bud by asexual reproduction, so they make clones. We don't clone ourselves, we have sexual reproduction, so we make gametes, and then the gametes come together in various genetically diverse ways, and so our offspring are actually genetically diverse from us, but look similar to us often. So the units of heredity are genes. They're packaged into all the chromosomes that we've already talked about when we went through mitosis. Humans are created by the fusion of two cells, or the two gametes, sperm and eggs. And you can just say eggs. Really, it's after ovulation, and when it gets fertilized, we say it's an egg. But oocytes, eggs, it's all the same thing. I usually will just call them sperm and eggs. The fusion creates the first somatic cell. Whenever you see the word somatic, it just refers to kind of the body. So there's reproductive cells and there's somatic cells. That means cell of the body. The first somatic cell of the embryo is the zygote, or the one-cell embryo, the zygote. And then what happens after that? What happens after you have that first sperm and egg unite, you have the zygote, that first somatic cell, and then what happens? Lots of mitosis happens, right? So lots of mitosis, so it divides and divides and divides and divides, and then you have all these different complex series of events that make up the embryo, and then make up the infant and the baby and the child and the adolescent and the adult, and that whole developmental series that we go through. So human sexual reproduction, we get the genes from two individuals. They're combined in random ways to make a new individual. So you'll have the one successful sperm fertilizing the egg, and then those genes delivered from the sperm and from the egg create this new combination of genes in this new unique individual. So we've got genetic variation and adaptability to a changing world. In other words, we sexually reproduce, and we'll see in a little bit, even our gametes making sperm and making oocytes. Those genes and chromosomes, they shuffle, they recombine, they shuffle and they change. And then you have a whole bunch of different gametes. So it's this whole process of massive variation of which one is actually going to come together. So the genes are absolutely unique. There's nobody else like you in the entire world. Even if you believe in like doppelgangers and funny stuff like that. You will not have the genetic combination that is you. And it's tremendously variant. And the cool thing is we have a world that's changing, right? Our environment from the dawn of time has been changing with weather, with climate. We've got global warming and all kinds of chaos right now. There's a lot of patterns and things that have shifted and changed. If you reproduce sexually and make very diverse, genetically different gametes that come together in different genetic ways and make brand new shuffling combinations, are you better able or worse able to respond to changes in the environment?
Better.
You're better able. Why are you better able to? A group of animals or people or whatever that reproduce sexually. Why?
Because of adaptation.
Because of adaptation. Okay, so adaptation, so traits or features that give you an added advantage, right? So if we have variation, so if I just make clones of myself and, I don't know, something happens where I need to be short, like short individuals survive. This is weird. I'm just coming up with this right now. Okay, bear with me. You need to be short, and I'm really tall, and all of my offspring are just like me. They're all clones. We have no genetic variation. We just pop off. We're all exactly the same. Catastrophe happens, and all the tall people are wiped out. I'm gone. All my offspring, I know it's weird, it's laughable, right? Okay, this is a bad example, but it's dramatic. So I'm gone, all my offspring, they're gone. But the individuals that actually have genetic variation, where their genes shuffle and combine in different ways, they will have a wide repertoire of different traits, okay? And those traits individually will be selected for. So say, okay, if you're shorter on the spectrum and you survive in that situation, there you go. Now you are gonna pass those genes on to your offspring. They're going to have those traits. They're going to survive. Those are going to be passed on to their offspring and so forth. You see that? So over time, certain traits and genes get selected for because they give survival advantage. It's really the principle of evolution, natural selection by evolution. It's if you have a whole series of genetic variants and even mutations, sometimes mutations arise and they give you an added advantage, then you will be able to have successful offspring. And we'll look later at chemistry and attraction and sex and love and all those things and how people come together, and even that is related to sort of natural selection and evolution and having healthy offspring. So we'll talk all about that a little bit more later. Was there a question about this specifically? Okay. So germ cells or gametes. So a lot of times people will refer to these as germ cells. Doesn't mean like you, gross germ, cold, flu. It's just germ refers to like early, nascent. These are the early cells, the gametes, the sperm and the oocytes form in the gonads through meiosis, and they have half the number of chromosomes. So because the sperm and the eggs, when they're finally formed in their full-fledged oocytes and sperm, they actually have half the chromosome number. So they are haploid. Ploidy refers to the chromosome number. So when you see that ploidy, this is sort of the chromosome number. And when you see half, that means half the normal number of chromosomes. And when you see diploid, that means sort of the normal set of chromosomes, or the double set of chromosomes. So here we have, and I spelled that wrong, there's only one P. Haploid is half the normal number of chromosomes. So how many chromosomes do human beings have?
26.
We have 26 pairs. 23 pairs. Actually, you were off. We have 23 pairs. We have 46 total. Okay? Yes. So we have 23 from mama. We have 23 from papa. And that makes 46 pairs. Okay, so you got to remember that. That's how many chromosomes. Mice, worms, flies, sometimes some little simple creatures have more than that. They have 58. Some plants have like 90 chromosome pairs. Some have 68. We have 46, 23 pairs, okay? So the sperm and eggs are haploid. They undergo a division, a reduction division, where their chromosomes are duplicated and then torn apart twice and reduced in half. So you don't have a sperm that has 46 chromosomes and an egg that has 46, and then you have what, like 92? No, we're supposed to have 46 total, 23 pairs. So that's why the gametes have this unique division called meiosis. Egg and sperm fuse, they restore the original chromosome number. The original number is diploid, a pair, one from mom, one from dad. So you got 23 pairs. And this activates the embryonic genome, okay? So the new individual progresses in developmental stages from zygote, the one-cell embryo, to the fetus, to the baby, to the child, the adult, right? Going through a series of stages, the life cycle. Each zygote gets 23 chromosomes from mom, 23 from dad. That makes 23 pairs of what we call homologous chromosomes. That's the diploid number, 23 pairs or 46 total. Homologous chromosomes are chromosomes that are pretty much the same, but one came from mom, you inherited one from mom, and you inherited one from dad. So they have the same series of genes on them, but they may have different mutations or different alleles. Brown eye color I got over here, maybe I got blue over here. A lot of those, we have dominant situations or recessive situations, so based on those genetic interplays will determine what protein or what color or what trait you have. But basically you have those 23 pairs of homologous chromosomes in you. So the 22 of those are autosomal. 22 are autosomal. That means they don't determine the sex of an individual. They determine everything all about you, even stuff to do with reproductive development and gonads and height and proportions and genetics of different traits. Those are coming from those 22 autosomal chromosomes. They've got all kinds of proteins they code for, many, many genes that code for different things. They have the same but not identical genes on them, okay? Then that last pair, that 23rd pair, are the sex chromosomes, okay? The sex chromosomes are X and Y. Those have mostly different genes on them. We'll see in just a minute. The X has a whole bunch of genes. The Y has just not as many genes. And they have certain ones that are similar genes, but they also have very different. Okay, so they're unique. They're uniquely identified to the X and Y chromosome. And they lead to the development of testes or ovaries. So specifically the sex chromosome will tell the developing embryo of whether to start developing testes or ovaries. And we'll talk more about that later, exactly how that happens. So if we look at these, we have the X chromosome, big fat chromosome, we've got the Y, okay, little short arms, long arms of the X, and then the Y. So the Y is very much smaller. It's smaller in size. Females have 2 homologous X chromosomes. So if this were a female, it would be 2 of these, okay, 2 X chromosomes. And if it's a male, it will be a Y and an X. Yes? We already know that, right? Most of you already know that, right? Females, those sex chromosomes, females have 2 X's, males have an X and a Y. Females will always pass on an X. So in other words, in all of the females in this room, ovaries, you will have your eggs, your oocytes, that will all have X's because you are XX. You can't make any other gametes that have a Y because you have all X's. What does that mean? It means that the male is the one that determines the sex. Their gametes, the sperm, determine whether they deliver an a sperm that has an X in it or a sperm that has a Y in it, okay? So males can pass on either X or Y. So the sex of the child is ultimately determined by the winning sperm. So I always tell my friends and family and stuff like that when the man is irritated, like, why I wanted a boy? I'm like, you're the one that determined that. They're like, no, not. Yeah, you are. Your gametes are the only ones that can give either that Y or that X. So sex determination comes from gamete of the male, okay? The X and Y look different, as I said, and they have different genes on them. The X has 1,100, closer to 1,200 genes total, all the way along the DNA that's all wrapped up into the X chromosome, condensed chromosome. The Y has only 80 genes. So very, very different. Not to say those are less important. They're important. We're going to see there's some critical genes on there that allow for development of the gonads or the testes and going down the boy path. But the X also has some very important genes that are involved in ovarian development and going down the female trajectory or development. Okay, so the overall life cycle, we spend very little time in this haploid state, right, where our gametes are actually haploid. You have egg and you have sperm. Now when they come together and they unite at conception, you get a restored diploid chromosome number, you get the zygote, so you have a 2N. So now the haploid state is just N equals 23 chromosomes. That's the haploid state. 2N is, I double that. 2N is the diploid state. I have 46 chromosomes. So all of you more than likely have 46 chromosomes. And I say more than likely because there are some variants and syndromes and different things where you have one too many or one too few chromosomes. So in general, individuals will have 46 individual chromosomes. Then we have mitosis and development of the embryo that gives rise to the baby and then the adult. Even in the baby, the embryo and the fetus, the ovaries and testes are already developing. Matter of fact, in the female, meiosis, the eggs are already undergoing meiosis in the fetal ovary. So they're already entering that gamete formation process in utero or in the development of the embryo. So meiosis is only happening in the gonads to produce the haploid gametes, okay? Meiosis is distinctly different from mitosis. Remember, we went all over mitosis. Mitosis, duplicate the chromosomes, rip them in half, so now you have the exact number that you started with, right? Because they replicate and then they divide, so they stay 2N. In the gametes, in the gonads, they duplicate, but then they rip in half and then they rip in half again. Okay, so there's 2 divisions, and we're going to go through that, and that makes them ultimately haploid, so that you can restore the overall chromosome complement to 46 chromosomes upon fertilization. We can look at chromosomes by karyotype, and many people will do this for genetic testing of different types. We can, in the cells when they're in a certain phase of mitosis, so they're dividing, usually it's at metaphase or prophase, the chromosomes are condensed, because remember, we don't want big squiggly noodles all over the place at interphase, the DNA's everywhere. The cell's gonna package up and wrap up its DNA in a tight, full-fledged chromosomes. And when they're in this phase, we can basically split open the cell, drop it on a slide, and use computer software programs to grab and put sort of electronically put together all the different chromosomes. So we can count how many. There should be 2 of each one. There should be 2 of number 1, 2 of number 2, 2 of number 3, 4, 5, all the way to the 22nd autosome. So the autosome chromosomes, number 22, and then the sex chromosomes, X and Y. This happens to be from a male. It's a normal male karyotype. So now if we look at these, we've got, they're kind of bundled up tight together, but we've got those homologous chromosomes. One is from mom, one is from dad. They duplicated though, so if you look closely, there's actually 2 connected at the centromere. The centromere is right here in the middle of these. So really, here's one like this, duplicated. Here's another one like this. They're just lined up, they're kind of holding on tight to each other. So there's actually 4 copies of chromosomal DNA present, 2 identical sister chromatids. So this one replicated right before mitosis. This one from dad replicated right before mitosis. So you technically have 4 copies of an individual gene or an individual set of alleles, right? So you have 2 homologous chromosomes for all the autosomal chromosomes, and then you have the X and the Y, or an X and an X if it's a female. So all this can be done really simply. And this is done as some of the first things for chromosomal and genetic testing and analyses. You can look at a person's chromosome ploidy or complement. I will say this kind of jumps ahead to pregnancy and miscarriage and stuff we're going to talk about later. But the number 1 cause of miscarriage in human beings and women is abnormal chromosome number, or something we call, I'll just add right up here, aneuploidy means inappropriate chromosome number, or inaccurate, wrong, I'll just write wrong, chromosome number. Wrong meaning the atypical, not the 46. That means I'm, say for example, 46XXX. I have an extra X, so technically 47 total chromosomes. Or monosomy X, I have 45. So I would have 44 autosomal, or 44 autosomal and only 1 sex chromosome. So there are individuals like that, where that we consider that aneuploidy. It's not the normal diploid chromosome complement. I'm sure all of you guys have heard, and in here we'll talk about trisomy 21. There's 3 of number chromosome 21, which is right here. There would be a 3rd additional one right there. Trisomy means 3 chromosomes for the number 21 chromosome, okay? So autosomal and sex chromosomes, we can analyze this. There's a lot of ways to do it. So how does meiosis work? As I said, you've got your pair of homologous chromosomes. They replicate. We're going to keep it simple. We're going to say sort of gender biased here. We're going to say like the red is the female and the blue is the male. So this is the maternal chromosome. This is paternal, coming from mom, coming from dad. Just keep it simple. They replicate, so now you have 2 identical sister chromatids from dad, and you have 2 identical sister chromatids from mom. Homologous chromosomes, in this situation, this homologous pair, homologous means they're very similar, right? They line up next to each other, and we're going to see they do something really, really interesting. They swap pieces of themselves. They actually have enzymes in your cells that cut, snip, snip, and put them together like a little jigsaw puzzle. So these get scrambled up a little bit, then they separate, they get ripped apart. This is the first meiotic division, or meiosis 1, okay? They go into 2 separate daughter cells. These have another round of division where the sister chromatids get pulled apart at the centromere, and they get pulled apart into their individual daughter cells. In this case, here's the maternal chromosomes went here and here, paternal ones went here and here, okay? So now this chromosome complement is haploid. So we just form either sperm or eggs by doing this process. Does that make sense? Okay. So, and it's different than mitosis, but it's similar, right? It is similar. This actually we see in mitosis, right, where they separate. So there's many similar features of meiosis and mitosis. It's a cell dividing and making daughter cells. In the case of meiosis, our daughter cells are either sperm or eggs, the very, very important cells that deliver the genetic material and make the next generation, right? In this meiosis, though, there's an additional round of chromosome segregation and cell division. So there's a second round of division. So there's 2. So you start with 1 and you end with 4. How many mitosis do you start with and end with when a cell undergoes mitosis? You start with 1 and you end with 2. That's it. So this is an additional division. These daughter cells go, we're not done, we're going to divide some more. Results in haploid genetically distinct gametes, and that is key. These are genetically different than anything you started with, right? And this is why I know some of you guys probably look more like your dad or look more like your mom, right? Or you have siblings and your siblings look really similar or nothing alike. We're going to see exactly why that is and the way that these shuffle, the genes actually shuffle. And they do that in this process called genetic recombination. This is called homologous recombination or genetic recombination. Crossing over is the simple way to say it. Crossing over, these chromosomes, these homologous chromosomes, at its first part of meiosis in prophase, they actually line up, they find each other, the proper proteins and enzymes and microtubules lining everything up. They line up, they find each other, and then this is where they swap or they cross over genetic material. Scientifically speaking, we call it chiasmata. Chiasma or chiasmata means to cross. So they make an X, they actually crisscross. There are enzymes that snip, 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 and they cross those 2 segments. In this case, there's a yellow and purple or pink and blue. They basically cross each other, swap those sections of those arms. In this case, here's the long arm, here's the short arm of the chromatids. They swap, so they end with exchanged material. You see that? So one was all maternal chromatid or chromosome. Now it's mostly maternal, but it's got some paternal chromosome on the end. You just made a brand new genetic combination that never existed before. You see that? So this is a really important part of meiosis, and this happens front and center in prophase at the very beginning. So you get chiasma or chiasmata, crisscrossing. Enzymes actually cut, snip, glue them back together. They get ligated. So now you have a piece of what was once like maternal chromosome on the paternal chromosome, and vice versa for the other one. You swapped. Those chromosomes are not going to stay together. They're going to go off into different gametes. So you just made brand new genetic combinations by doing chiasmata. This is from your textbook. It's not a clear figure. It turned out kind of washed out, but it's demonstrating the same thing. And I will say, it doesn't just happen once. It typically happens 1, 2, 3, 4, 5, 10 times, all over the place in the different chromosomes. So it's reshuffling, genetically recombining the genomes of those 2 different sets of chromosomes. Yes, Sarah?
When would this occur, like when the child is born?
No, this is happening in the gametes, in the gonads, when meiosis is happening. Every single one of your gametes, and actually in the female, this is already happening in the fetus. Your fetus happened a long time ago. It's already happening. In the male, it's happening all the time as the gametes are being produced throughout sexual maturation and later life. So meiosis is forming your gametes. That's long ago. So they shuffle, they combine, and then they wait for the right signals to continue the process of meiosis and give full-fledged eggs. And in the case of sperm, that's going to happen throughout the male's life because they have stem cells, so they can reproduce and make sperm to the time they're 90. We'll talk more about that later. But the mechanisms of meiosis is what I want to get across right now, okay? So this is happening in your gonads, in your gametes. Females, it's already starting back in the fetus, so it's already happened for most of you, already a long time ago, crossing over, snip, 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 exchange. And in male gametes, it's happening as they produce sperm throughout their mature sexual reproduction life. So we have chiasmata happening. This is meiosis 1, prophase 1. Tetrads, we call these tetrads because it's a set of 4, right? It's the 2 pairs of homologous chromosomes. The 2 chromatids from mom, the 2 from dad, they line up, they get sort of glued together temporarily, and then enzymes help to cut and snip and exchange material. Now notice this, a lot of the textbooks will keep this simple. They'll just show 3, because showing 23 sets of chromosomes doing this would be a nightmare. So I'll use 1 or 2 or 3 just to illustrate, but remember there's 23 sets of these homologous pairs. Then the centrioles grow out the microtubules, right? And so they finally position the poles where you have the mitotic spindle. Those tetrads get grabbed and they line up along the metaphase plate. Okay, you already know all this. We did this already, right? Metaphase plate, this is metaphase. Then they get ripped apart. There's enzymes and motor proteins that are anchored to the chromosomes and walking on the microtubules and are kind of doing a little tug of war and pulling them, and they get pulled to the two different poles. Now the interesting thing in meiosis, this is very different than mitosis. You've got genetic recombination here. You've got chiasmata to exchange pieces of those chromosomes. That's very unique. This increases genetic diversity and uniqueness of the gamete that's going to form, right? But we also have this process right here, and I believe I have an animation. No, not really anything fancy. Independent assortment. What we call this is independent assortment, and that is the maternal or red chromosomes and the paternal or the blue chromosomes. They randomly end up on different sides of the equatorial plate. Okay? So they find each other, they grab each other, and then they get grabbed by the microtubules and they get pushed up and line up along the middle, get ready to be ripped apart. But notice how they pair. You've got two maternal on this side and one maternal on that side. And then two paternal over here and one over there. So this process of how they get positioned when they line up with respect to the two different poles is independent and random. We call this independent assortment. So I will say right here, genetic recombination, okay, and this independent assortment, these two things are what allow your gametes to be so incredibly, uniquely, genetically distinct. Because these things don't happen in mitosis, right? These are lining up in such a way that I could have more chromosomes from the maternal side in a particular gamete that's going to become me or an offspring of me, and less paternal chromosomes going to that side. And so you can see, if you get more of the maternal going to that daughter cell is going to have more genetics, alleles, that are similar to her mom. If you have more coming from the paternal chromosomes, you're going to have more traits and features in common with your dad. So this is independent and it's random. Okay, so genetic recombination, chiasmata, and independent assortment, how they line up randomly, the maternal and paternal chromosomes, helps to make sexual reproduction, not just in us, but in all sexually reproducing species, very genetically diverse. This is why there's nobody like you. This is why you have absolutely nobody else that you share the same genes with, unless you're an identical twin. Identical twins came from meiosis of a sperm, meiosis of an egg, genetic shuffling. They came together, and then that egg either split at the 1, 2, or 4 cell stage. So they have exactly the same genetic shuffling of material. So that egg, and we'll look at that when we talk about embryo development and pregnancy. We'll look at twins. But everybody else, if there's only one of you, even fraternal twins are just like regular brother and sister. They have their own genetic shuffling and their own separate sperm and egg. All right, so ultimately, actually I'll skip back one. Ultimately what happens at the end of meiosis 1, anaphase is pulling those genetically recombined chromosomes apart to the different poles, and now you've got two different daughter cells. Now the two different daughter cells, there's no more tetrads, there's no more homologous pairs, because they got pulled apart. Now it's almost like mitosis, where you just have your complete sister chromatid, right? And then they line up on the metaphase plate, they get pulled apart, and now remember, when it goes from sister chromatids that are basically duplicated chromosomes, and they get pulled apart at the centromere, that actually results in individual chromosomes in those different daughter cells, right? So we have individual chromosomes here, we have the set of 3 of them, and 3 of them, and then 3 of them, and 3 of them. So we just went down into now haploid daughter cells with telekinesis, cytokinesis, division of the cytoplasm, cytokinesis happening. So ultimately we're going to have 4 at the second round of meiosis. Okay, so first one you get 2, and then those go on and divide. Now you got 4, so you went from 1 to 4. I will say a little caveat here though. Male sperm going through this process, meiosis, will create 4 full-fledged sperm with meiosis. Female eggs, you will only end with 1 egg. You will end with 1 egg and 3 little skimpy little tiny little polar bodies. So in sperm, you will end with 4. But with eggs, you will end with 1 oocyte. And something basically the big main daughter cell with these little tiny little, sorry, pinched off chromosomes. And females put everything they have, all the proteins, the RNA, the enzymes, the mitochondria, all that is chock full and is put into one gamete. And then when it undergoes meiosis, it just pinches off the tiny little cells that we call polar bodies. We'll talk more about, these are polar bodies, we'll talk more about these later. But when we get to female and male reproductive anatomy and puberty and all that stuff, which is coming up. So oocyte, you only end up with one. Even though it went through the same meiotic divisions, it just pinches them off into little small ones. It goes, nope, we need to be nice and big. The egg is the biggest cell in the entire body. It's over 100 microns. It needs to be full of all the proteins and material and mitochondria and stuff. But sperm are small, compact. They basically just have genetic material that they're supposed to deliver, so you end up with 4 sperm at the end of meiosis 2. Okay, this is kind of dark and hard to see, but summarizing what I just said, 4 sperm happens by the end of meiosis 2, and then you get 1 oocyte with 2 to 3 polar bodies that get pinched off and divide away from that egg. So 1 oocyte with 2 to 3 polar bodies, depending on if one of those polar bodies divides again and undergoes meiosis 2 itself. But ultimately, you get 1 good egg or 4 sperm from meiosis. Yeah, Simone.
Does chiasmata only happen during meiosis 1?
Yes. Chiasmata, or genetic recombination, only happens at prophase 1 of meiosis 1. We can call it chiasmata, crossing of the genetic material. Happens specifically in prophase 1. And this is how when you see like prophase 1 or prophase 2 or anaphase 1 or anaphase 2, you know that's referring to meiosis, because meiosis is the only one that has 2 rounds of cell dividing. So it goes prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis 1, and then prophase 2, metaphase 2, anaphase 2. Get it, right? Okay, so that only happens there, because that's the only time when those tetrads, those 2 pairs of homologous chromosomes, line up, find each other, and get bound. That's the only time they actually exchange, because after that they get pulled apart. Remember, right in anaphase 1, they're pulled apart, they're new daughter cells now. It can never happen again, okay? And actually something interesting going along with miscarriage and aneuploidy, wrong chromosome numbers, a lot of times you get wrong chromosome number if the chromosomes don't undergo genetic recombination. So if they don't do chiasmata and exchange pieces of the chromosomes, it almost is a trigger that those chromosomes are not going to split the right way. They're not going to come apart and go to 2 different cells. They're going to kind of stay together. So we know that non-disjunction, and I'm getting ahead a little bit, but if your chromosomes do something called non-disjunction, what do you think that means? What does it sound like? They don't separate, right? They don't disjoin. They don't come apart. They stay together. So you have chromosomes staying together so they don't come apart.